Good morning, students. Good morning, class. We are going to be going through Module 1, DNA, Genetics, and Invitation Notes. So if you want to go ahead and pull up your PowerPoint so that way you can follow along with me. And if you want to go ahead and get yourself set up before we go ahead and start taking the notes, that would be great. Just a reminder that these notes do need to be turned in through pictures on Canvas before the unit test. So it'll be done at the end of the unit test. So let's go ahead and get started. Okay. So we're going to go through the DNA invitation notes. So module 1, what is DNA and what is it made of? So this unit is really going to go into like the scientists behind DNA. What is DNA? Like, what is it made of? What is the structure and the basis of DNA? Okay. So Franklin and Wilkins. Usually you just hear about Rosalind Franklin, but the study that was actually done was actually by Rosalind Franklin and Maurice Wilkins. And they took the first picture of DNA using X-ray crystallography. Okay. This is when DNA is bombarded with X-rays. Okay. And it causes a pattern to be able to be captured on film. Okay. So you can see here that there is plenty of evidence for the structures that line up. And this is where they actually got their singular helix theory about DNA. So Watson and Crick utilized the X-ray images produced by Franklin and Wilkins to create a 3D model of the DNA molecule. They have the double helix theory. So Franklin and Wilkins took a picture of DNA and they hypothesized that it was a singular helix. So it was just a spiral that goes like this, right? And this was used by Watson and Crick without citing them as like a source, which is really important. This is actually kind of one of the things that like led up to the purpose of citations. And then they published this double helix model of DNA. Okay, what is DNA? We've talked a lot about like how it was discovered, but what is it? DNA is deoxyribonucleic acid, and it codes, so it has a code on it, and it codes for proteins and all the cellular activity, okay? What is DNA made of? So over here you can see there is an example of a DNA nucleotide, okay? So what is DNA made of? DNA is made up of nucleotides, so that's this whole molecule, okay? The monomers that make up DNA, okay? Those nucleotides then are broken down into 3 parts. This is the phosphate group, the deoxyribose sugar, so this is the pentose sugar, and then this is the nitrogenous base, okay? So in this case, this is adenine, which is one of the nucleotides that makes up DNA, but this all together makes up a nucleotide, but there are 3 different parts, okay? There are 4 types of nucleotides, okay? So here we can see the structure of a guanine nucleotide, okay? That's what G stands for. So you can see this is the phosphate group, this is the sugar, and then this is the nitrogenous base, okay? Sugar, phosphate group, nitrogenous base, okay? So guanine always pairs to cytosine, okay? And adenine always pairs to thymine, okay? So let's think about this. If there's 100 base pair section of DNA and there are 45 cytosines, okay? 45 cytosines means that there's going to be exactly 1 cytosine for every guanine, okay? So if there's 45 cytosines, there's 45 guanines, okay? So out of 100, if there's 45 cytosines, how many adenines are there, okay? If an 80 base pair section of DNA contains 30 adenines, okay, 30 adenines is a 1:1 ratio with thymines, okay? So 30 adenines to 30 thymines. Well, I just gave you the answer, but how many cytosines are there? Just think about that, okay? Double helix. Strands of DNA wind around each other. So as we can see that these are winding like this, okay? This is actually a really unique pattern to DNA. It's not seen a lot of other places in nature, okay? And they wind like this, and they fit together, and they're opposite of each other. They're complementary. You can see that the 2 different base pairings are always in their pairs. They're never not in their red and green or their yellow and blue pairs in this picture, okay? So the main bonds in DNA are hydrogen bonds. So hydrogen bonds are bonds that are holding the base pairs together, and it's actually how the nucleotides match up, okay? And then it's covalent bonds that hold the sugar and the phosphate together. So it's covalent bonds here, okay? And covalent bonds here. And then between each of the nucleotides, between the G and the C, there are hydrogen bonds. Hydrogen bonds are stronger than covalent bonds, okay? DNA and chromosomes, okay? So here you see a prokaryote and a eukaryote, okay? Now, in prokaryotic cells, DNA is located in the cytoplasm, okay? So it's just one big circular DNA molecule that contains all of the genetic information, okay? It's called a plasmid, circular DNA, okay? In eukaryotic cells have organ-bound membrane, organelles that are membrane, right? So, whereas prokaryotes don't. So in eukaryotic cells, DNA is located in the nucleus of the cells, okay? And then each organism, so every type of species, so humans are one, dogs are one, like cats, they each have a different number of chromosomes, okay? This is the structure of a chromosome. So a chromosome is just a lot of DNA, okay? DNA is tightly packed together to form chromatin. And chromatin consists of DNA coiled around histones. Histones are proteins. So this is the histone. This yellow thing is a histone. The DNA is coiled around it, okay? And that coiling coils even tighter, and it becomes this chromosome that we think of, okay? They're thick. They're very thick and fibrous, okay? So I want you to watch this recap video and take some notes on this. And then when you're done with these notes and you're done with the video, make sure you take a picture of your notes and you submit it to Canvas. That is all I have for you for DNA notes for Module 1 for the invitation. Just make sure that whatever notes you took, you take a picture of it and you submit that on Canvas. That way you get your points and you get your credit. Have a wonderful rest of your invitation.